numbers of people coming into our seminars. Just to note to everyone, the session is being recorded, so in principle, you can watch it later. Gerard has also generously offered to make his slides available later through the network, so you should be able to access that later on as well. I'm just going to admit one more person in. Gerard, are you ready?
I am ready.
Excellent. So you might want to start sharing your slides.
Sure.
Then I'll do the introductions.
All right.
Excellent. That's working. Okay. So good afternoon to everyone from the UKs and good morning to anyone coming from the US. We are back with the RSC Process Chemistry and Technology Group seminar programs. So today we have the pleasures of having Gerard Capellades, who's an assistant professor at Rowan University, who's going to talk about impurity and crystallization. So just to give you a quick introduction, Gerard is now an assistant professor at Rowan University in the Department of Chemical Engineering. He did his Ph.D. at the Technical University of Denmark, and then after finishing his Ph.D., he moved to MIT, working with Alan Meisens and Richard Brass. And his whole career has very much been focused on crystallizations in one way or another. In fact, in his PhD study, he designed a continuous crystallizer for productive APIs crystallizations. And his current research include very much everything about crystallization, so crystallization kinetics, molecular solid solutions, application of high throughput screening, digital twin for crystallization and process designs. And today, very much he will talk about impurity and how to deal with them when you have them in your process. So without further ado, I'm going to hand it over to Gerard. To the audience, if you got questions during Gerard's talk, please put them into the chat box and I will collect them and read them out at the end of the presentation. So over to you, Gerard.
All right, thank you so much, Bao, and thanks everyone for attending today. So as Bao introduced, we're going to be talking a bit about impurity rejection. That's one of the thrusts that we have in the group. I'm going to start first by introducing my group slightly. So we are actually located a little bit south of Philadelphia in the US. Yeah, we have a group that's mostly for grad students, postdoc, and several undergrads. We work a lot with students who are taking their bachelor's degree, who spend some time in the lab. I think that a lot of the work that is done on crystallization is something that fits very well into the training of a student who's doing their bachelor's, right? Even things like solvent screening and things like that. And when it comes to the type of work that we do, we have overall expertise based on like publications and stuff. We've been focusing a lot on impurity rejection solid solutions, right? I started with continuous manufacturing as Bao introduced, but currently we're really focusing on this topic, and it's almost every project in the lab works on this in one way or another. So let's start very simple, right? About sources of impurities and retention mechanisms that we've seen in literature, right? And then we're going to start bringing things a little bit more into shortlisting different mechanisms and trying to work a bit on diagnostics within that shortlist, right? So as many of you will be aware, and this is the example, for example, on organic synthesis and pharmaceutical synthesis, you have several steps involved, right? And in each of those steps, you're able to generate some potential impurities, right? You have a product of interest, in this case, this is synthesis for ciprofloxacin, but you also have a bunch of other compounds, right? That would be unreacted raw materials, intermediate products, synthesis by products, even solvents, right? That could be considered potential impurities, right? So there's a lot of work going on efficient synthesis for different pharmaceuticals, for different products, and a big portion of the research is also to improve things like selectivity. But ultimately, when you're going to design a crystallization process, you're most likely going to find a fairly complex mixture, even if the synthesis is fairly optimized, right? So to give you an example, these are the syntheses that we would find, or these the synthesis product that we would find for ciprofloxacin on the previous synthesis that I showed before, right? In which you pass that through the HPLC chromatogram, and you can see how there's, different many different impurities, right? On different levels, of course, depending on how the synthesis was conducted, right? And some of those impurities may be like in very low amounts, but can actually be fairly mighty on things like slowing down filtration or changing like the crude properties and all that sort of stuff, right? Now, the problem with that is you're trying to develop a crystallization process as a way to purify this material, right? So crystallization, we know that it takes advantage of this tightly packed, disorganized crystal lattice, right? That makes impurity incorporation not too favorable, right? It still happens, and that's why we have a career, why I have a research program, right? But in general, crystallization tends to be fairly efficient at separating different impurities from the product. So you can start with this crude material that comes from the synthesis. You can run a crystallization step, right? And then you're basically filtering the material. You recover the mother liquid that contains all the impurities, that goes aside, and then you can basically wash and dry your product, and you tend to have a fairly good purification efficiency, right? But it's not perfect. And the problem is in many fields like pharmaceuticals, you need higher levels of purification. And for that, you need to start understanding the impurities that actually made into the final product. How did they make it there? And how do we get them out next time, right? So if you look at how process development is done with impurities in mind, it's actually fairly iterative and it's fairly based on trial and error. I mean, we know things like solvent choice is going to be one of the main drivers in this, right? And that's a big portion of what we focus the screening on. But there's a little bit of like what we had to deal with in this particular project, which kind of inspired my research program later on, right? You start with like try to identify like, okay, which impurities are intermediates, can we improve the reactions, all that sort of stuff, right? And then you try different crystallization conditions, primarily different crystallization solvents, different wash solvents, to try to figure out, what is the best way to remove those impurities that are more challenging. Ultimately, you always end up with surprises, especially during scale up, right? And that tends to going back and doing some additional screenings and tweaking some of the crystallization conditions, right? And while this may be an effective way to design a purification process, it's not always the more safe option, right? You're trying different solvents, you're trying different um, your outcomes that you're measuring is basically how much of an impurity ended up in the final product. And you don't know if that impurity ended up like that because it was, let's say, supersaturated, but with a long induction time in your experiment, and therefore it never precipitated at the smaller scale, right? And then you're now scaling things up, and suddenly that impurity is able to crystallize because it has a much shorter induction time, and suddenly the impurity that was not showing up in your early process development is now appearing, right? Um, or there could be additional mechanisms by which the impurity may end up inside the crystal lattice, right? And that's one thing that's a bit more difficult to control, right? So the question on this is, instead of, trying and testing different crystallization conditions based on heavily on intuition and knowledge that solvents are a big factor into this, can we try to exploit mechanistic understanding on how each of those impurities is actually ending up inside the crystalline powder? And can we utilize the mechanistic understanding to make process design strategies that are scalable, right? That are based on thermodynamic control rather than empirical measurements, right? So when you look at literature, there's several impurity detection mechanisms that have been identified for how some impurities may end up in crystallized powders, right? In crystallized and filtered powders. So one of them is, and I like to classify them in blocks, right? So one block would be different forms of lattice incorporation, right? Let's say the impurities are inside the crystalline lattice, without being in solution, like basically independent units of the impurity or blocks of different impurity molecules, but without being an independent solution, right? And within that, you can see different mechanisms. That's basically my review on literature. I think that some mechanisms are actually end up being the same thing. But there's mechanisms that are based on like adsorption and incorporation, solid solution formation, defect sites, and even the formation of co-crystals, right? Those are different ways in which the impurity molecules will end up inside the crystal lattice, right? Now, another way that it can end up within the crystals or inside the crystals, but not integrated within the lattice, is in the trapment of mother liquor within powders or within crystals, right? So you can have examples of solution pockets that are formed inside, growing crystals. Those are fairly rare. We're going to discuss that in a moment, right? But you can have solution pockets, and of course, if your mother liquor contains all those impurities, the crystal will end up containing that, right? And same goes with things like agglomeration, right? You start agglomerating all those crystals. You make hard-bound agglomerates where the wash solvent is not able to penetrate within, and you end up having some other liquid that's trapped in there that basically contributes to the impurity levels in your powder, right? Other options is what I like to call externally, right? So in this case, it's either the impurities precipitating or crystallizing next to the product, forming its own lattice, right? And then you end up with a mixture of the two different compounds. Or it could just be a poor washing step in which you basically have some adhering solution, adhering mother liquor, that is essentially there on that contains all the impurities. And as soon as you dry that powder, if you're not washing it properly, it's gonna end up, precipitating and staying in the final powder, right? Finally, another mechanism that has quite a bit of popularity, but we're going to discuss that in more detail later, is surface adsorption, right? What happens if I basically add my crystals, pure crystals in solution, or if they grow with relatively high purities and then the impurities slowly adsorbed in the crystal surface and that contributed to incorporation, right? Now, of course, you have several mechanisms involved in here. And if you were trying to investigate each and every one of them, you would actually spend like multiple careers and also it would not be the most efficient thing to do in process development, right? Because now you have to test for every specific mechanism that's available in there. So one thing that we ended up doing is we came together with the teams at Boehringer Ingelheim and Merck, and we posed this, a simple question, like which mechanisms are first of all common, right? And which can also be responsible for impurity entrapments that are concerning for, let's say, the pharmaceutical industry. So something like would be like 0.1% or higher in the final product, right? And when you pose this simple question, you realize that a lot of these things can be simplified, right? So co-crystals, if you accidentally form a co-crystal between your product and an impurity, you will see that immediately on the X-ray pattern, right? And it's also very, very uncommon. Things like mother liquor inclusion, solution pockets, like there's very, very few crystals within powder. Even when you crystallize them really poorly, there's very few crystals that actually contain meaningful solution pockets. And keep in mind that impurities in liquid, in mother liquors are typically at the order of like 1 gram per liter or lower, right? That is 0.1% lower, right? So you're dealing with, or yeah, 0.1% or lower. So you're dealing with a solution that's only a small percentage of the overall crystalline phase, from which the impurity is only a very small percentage of that solution. If you do the mass balance, and we did that in a separate publication, if you do the mass balance, you realize that you need very extreme cases for those to actually cause significant level of impurity entrapment. Same goes with surface adsorption, right? You have crystals at the order of 10 to 100 micrometers. You can cover the whole crystal with impurities, and it's still a tiny, tiny portion of the overall crystal mass that's actually going to be covered by impurities, right? And then when you go to other things like adhering solutions, yes, that is a problem, especially if the washing is not done properly. But because you always end up drying the product, right? You end up precipitating it, so it turns into the other mechanism, right? It ends up turning into the impurities now crashing out, either because you washed it with a solvent that was making it precipitate, or because it just, you evaporated the solvent, it dried, and then it crashed out on top of the product, right? And you're left with basically two types of impurity retention, right? One of them is, well, the one when impurity is basically crashing out on its own. And the other one is a form of lattice incorporation, a form of non-stoichiometric lattice incorporation. Now, in here, we can have some discussions, and I don't want to spend too much time on this, but we could have some discussions about whether the adsorption and incorporation are really just a mechanism to make a solid solution, that the impurity needs to attach to the surface before it can integrate into the lattice, right? As I said, this is more a review of literature that I did, more than my own opinions on this. But ultimately, you end up with a form of non-stoichiometric lattice incorporation, right? That's the practical perspective. So what we end up doing in here is we work with these two groups, right? With the group at BI and the group at Merck. And we basically grabbed a bunch of, first of all, model compounds that anyone could reproduce, and that was what my lab was doing, right? But we also grabbed primarily every pharmaceutical, every intermediate that had caused some challenges with impurities at BI and Merck in recent years. And we ended up getting with 52 different compounds, 52 different pairs of product impurity. And we tested what impurity retention mechanism they were facing. And the interesting thing is that 79% really were able to form some form of solid state miscibility that was detectable between product and impurity, forming these solid solutions, right? This non-stoichiometric lattice incorporation. And then 21%, of course, the ones that cause a lot of challenges tend to be the impurities that precipitate, right? There was a 27% if you count miscible precipitates. That was basically that precipitation of impurity that caused very, very high levels in the product, right? Now, solid solutions was a bit surprising to see that because it's not really a topic that you see a lot in crystallization literature for organic molecules, right? But they're actually not a new topic in society, in nature, right? So you look at metallurgy, right? And steel is basically a solid solution, right? Bronze is a solid solution. Those are things that we've been using for thousands of years, right? You look at minerals, you look at most rocks on the street, right? They are also incorporating impurities and gemstones, right? They owe their color to an impurity that's incorporated usually at very, very small levels in the crystal lattice, right? You look at biological systems, that's some recent work, working on like guanine and hypoxanthine solid solutions forming zebrafish, right? Those are biogenic crystals. But you also see that in nature, some organisms actually use solid solutions to regulate biological functions, right? And the truth is, like, nothing is pure in either nature or manufacturing, right? We tend to simplify things a lot when it comes to in chemistry, right? And we tend to call systems insoluble or immiscible, right? You look for the solubilities of curcumin in water, and it's going to say, well, it's insoluble. It's like, well, no. I put some curcumin in that water, and even though the solubility is like 10 to the minus 5 grams per liter, I'm still going to get some staining of that water with the curcumin. There's some going into solution, right? You mix water and heptane and you say, well, those are not miscible, but you put them together and you'll realize that there's some heptane in the aqueous solution and there's some water in the heptane, right? The same happens at the solid state. Entropy is not going to let a crystal that contains 0 molecules of a foreign species if it's grown in the presence of the foreign species, right? And what you end up seeing when it comes to these organic systems, to these a bit more complex crystalline structures, is that you don't have this full miscibility, but you have this partial miscibility instead, right? So you always have a miscibility limit. Sometimes it's at the order of like 0.1%, 0.5%, sometimes it's at the order of 10%, right? But part of what we did to try to prove this is our collaborator from BI, like Fred, basically got one of my PhD students as a co-op and another PhD student from Stevens, and basically they started building phase diagrams, right? They started grabbing different impurities on salicylic acid, and they started building phase diagrams to basically show that it's not that different from what you see in metallurgy, right? That at the moment you understand these thermodynamics, you can actually understand impurity rejection much better, right? So, of course, this has a lot of implications, right? One of the things that you can already see in here is like changes in the melting point, right? You see those on the phase diagram already. You can see changes in the melting point of the mixture whenever you have impurity. You can also see things effects on solubility, right? So these are effects on pure API versus API containing impurities, where you can basically see one of the industrial compounds, I believe from Merck. And you can basically see at the moment you add a small amount of impurity, depending on the solvent, you get different levels of solvent enhancement, right? Those in process development are challenging because they lead to lower yields, right? So you can have this case, that's the ciprofloxacin that I presented at the very beginning, where we typically get with commercial material close to 89% yields on continuous crystallization, and then through a combination of growth inhibition and also solvent enhancement, you would actually end up getting a 73% yield at the moment you add 3% impurities in there, right? Now, this is also an opportunity. So this is an example of from the University of Limerick, from Matteo Lussi's group, in which they were basically forming solid solutions of praziquantel with malic acid and tartaric acid. And one of the things that you can see is significant sorbate enhancements and faster drug dissolution. So those could be really an alternative to co-crystals when it comes to solid form design, right? So what is a challenge in process development, it could also be an opportunity for product design in itself, right? Now, the other thing that you can notice is if those solid solutions are also able to affect solubility, right? And their miscibility is, of course, going to depend on the crystalline structure of the host. You can actually destabilize certain stable forms with impurities, right? That's something you can already see in some of these phase diagrams that I showed before. But you can actually see how, for example, in 2,3 dihydroxybenzoic acid, the pure material will always be crystallizing in form 1, but at the moment you add a little bit of salicylic acid into that, you start getting a coexistence domain between forms 1 and 2, and eventually you end up making form 2. And if you measure the solubility enhancements on those, what you can see is that form 1 is getting a significant increase in solubility, right? It's being destabilized significantly by the presence of impurity, much more than form 2 is, right? And eventually you end up having this switch. There's also some computational work, also supported by experimental work from the University of Durham, where they were basically also doing the same thing with nicotinamide and benzamide, right? So we're starting to see a bit on like, you know, the issues with disappearing polymorphs and the role of impurities on changing crystallization. We're starting to see a slightly new lens on trying to explain this, right? Now, one of the questions that I would have when I see all these papers and when I, you know, work with these collaborators that we're mostly working with, like, you know, a white powder mixing with a white powder impurity, right? And one of the things that you notice is when you go back to metallurgy, kinetics are very important. Kinetics are going to be defining the overall spread of, let's say, like, you know, carbon within lattice of steel, right? And in that case, I wanted to see if you have this sort of like gradual miscibility of one product on the impurity in the product. Can you actually have also gradients of impurity within a single crystal? And how does this impact everything? How does this impact like solubilities within a single crystal, right? So we decided to add a little bit of color on research, right? We started looking at literature and we find some dyed crystals, of course. If you can have an impurity that has some color, even though it's not the most industrially relevant, it allows you to fundamentally study how the systems behave, right? So this is an example for potassium sulfate. And if you mix it with acid fuchsin, you would initially think that a very large molecule like this is not going to fit into the potassium sulfate lattice, but it actually fits fairly well, and it fits in a fairly substitutional manner, right? That's the interesting thing. If you were checking that based on, like, you're trying to measure the impurity content based on, let's say, HPLC, you would not be able to find it unless you're really looking for that, right? But it's miscible. It's there at ppm levels, right? But it is there, and it has some effects, including things of morphology, right? So, and the idea here is like, this is not like the fact that it's forming this sort of hourglass. It's not a consequence of the lattice being any different in one location over the other, right? This is simply a kinetic effect, right? Crystals have different surface properties. They are anisotropic by design, right? So they have different surface properties that can potentially interact with certain phases, interact with impurities preferentially, right? But if you put that into temperature cycling, you can basically slowly have this homogenization of these crystals, right? You can basically prove that there's some miscibility throughout the entire lattice, and that the reason why they are forming these sort of hourglasses is really kinetics, right? It's the growth history of that material, right? You can also see that on things, crystals that regenerate, right? You have a crystal, you chip it. This was a case that happened by accident, where a student of mine basically grabbed it a bit too hard with tweezers and make some like fracture areas, right? And after putting it back in an impure solution, guess what? If the crystal is regenerating in an impure solution, those areas will contain impurity preferentially, right? So there's few things that depend on growth history, kinetics of crystallization, as well as the overall fracture, regeneration, things like that, that affect how the impurities distribute in the crystal, right? If you compare the hourglass crystals that we grow following Carr's methods on K2SO4 and acid fuchsin, low supersaturations, no mixing, just low evaporation or petrities, you get these very nice hourglasses, right? But if you do the same in a stirred tank, suddenly you're getting very, very different morphologies with very, very different locations of impurities. Not even an hourglass anymore. Now it's like clean tips and an overall impure center core, right? Which could potentially be faster crystallization at the beginning, right? Or like, higher supersaturations leading to more incorporation, or it could be basically a loop of like breakage and regeneration. We're still investigating all of that, right? So we try to find also our own systems, right? We don't want to be stuck only with K2SO4. Can we do the same with pharmaceuticals to kind of show that it's the same? So we grabbed the most common pharmaceutical, like paracetamol, that you would see in like most crystallization papers when it comes to pharma, right? And we tried the common dyes that you see in all these popular dyed crystals literature, right? So we grab acid fuchsin, of course, and of course, acid fuchsin incorporates inside acetaminophen. You grab curcumin, right? And it's also incorporating and forming sometimes very, very interesting hourglasses, right? They're much more difficult to get than K2SO4, but they sometimes are forming like this. And then sulforadamine B, which is another common dye on this dye crystals literature, and you can also see that it incorporates also at variable levels within the lattice, right? Other things that you can notice in here is those are like parts per million incorporation. At most, we get like 500 ppm, 600 ppm right now, right? It's very difficult to measure at some point because the crystals also become very fragile when you try to grow a single crystal in large amounts, and powder gets dirty with impurities, and it's a bit more difficult also to it gets dirty with solution that contains a lot of impurities. But things that you can see is that if you bring those impurity levels overall to something like 80%, you can have significant solvent enhancements, right, with very, very limited lattice incorporation. Now, some of these solvent enhancements are a function of the impurities in the liquid, right, not just the solid state. But there is significant effects on the solid state, and one way that you can see that is on melting points and enthalpies of melting, right? So you can see how small levels of something like acid fuchsin can actually drop the melting point of acetaminophen, and it can also drop the enthalpy of melting, right? So the question in here is that if we put two and two together, does composition and anisotropy also correlate with the solubility effects that we were seeing before, right? We see higher solubilities on impure crystals, and you see faster crystallization kinetics on the crystals that contain impurities. Can a single crystal present like a gradient of solubilities depending on its purity, and can it dissolve at different rates directed by the location of those impurities, right? So at that point, it was early on in the lab, right? We didn't have more than a phone camera and a sink strainer. So we basically grew the crystals in that, and we basically put it in a sink strainer and took pictures of the phone camera on how this is the case for acetaminophen and curcumin, how it dissolved in water, right? And one thing that you can see is that after a while, the impure regions are gone, right? It's surprising because acetaminophen solubility in water is like 6 orders of magnitude larger than the curcumin solubility. But this is not about the affinity of the impurity for the solvent. This is a comparison between a lattice that is pure and undisturbed with a lattice that contains the impurity. And that's one system where the solubility enhancements were very, very difficult to see. acetaminophen sulforamine B. So my student got like very frustrated at this, like what if it could be a consequence of one phase dissolving faster than the other, right? So she grabbed the crystal that contained the most random distribution of impurities, placed it in water, and of course, it also ended up removing the impurities first, right? Potassium sulfate acid fuchsin, the famous system, same thing, right? Eventually, we ended up getting a better camera, and we basically recovered the crystal like midway through dissolution, took some pictures with a new microscope, right? And the one thing you can notice in here is like, yeah, the impurity regions being eaten away with the solvent, right? During that dissolution stage. We also started taking snapshots over time on how that dissolution is proceeding in some of those crystals. And again, you can see pure crystal, crystal with impurities. There's a very, very different dissolution profile that's going on at the moment you have impurities incorporated in the lattice, right? And those are, again, K2SO4 and acid fuchsin, very, very limited effects on bulk solubility. It's very, very difficult to see, yet the impure regions tend to dissolve faster, right? Now, the interesting thing here is that I can go back to diagnostics, right? And I can try to get those images. I can basically measure the percentage of the crystal that's dyed and the overall surface area of the crystal. And I can model the dissolution progress, right? And one of the interesting things here is that you can, of course, see how you can dissolve a very, very small amount of the crystal, but you can lose most of the impurity within that dissolution, right? That helps explain why some reslurring and washing steps are so effective, right? It's not just about dissolving precipitated impurities, it could also be ripening some of these solid solutions, right? But it also helps explain some issues with diagnostics, because if you didn't know all of that I've been presenting now, if you didn't have those images, and you grab some powder and you dissolve 20% of that powder and 60% of the impurity leaves the powder, where would you think those impurities are? You would think those are surface adsorbed impurities, right? I think we've been misdiagnosing solid solutions, and that's why they've been absent from literature, because we tend to think that if I dissolve 20% of my material and most of the impurity comes out, it must be at the surface. It must have precipitated as a film on top of the surface. It must have attached itself with surface adsorption, but it doesn't have to be that way, right? It could be lattice incorporation that's being masked like this, right? All right, so implications for process design, right? I want to use the rest of the time in like, how do we deal with this? Now that we know how they behave, now that we know what they are, let's figure out diagnostics. So the one thing I want to bring to everyone's attention, basically, is the solubility limited impurity purge test, right? That's one that it wasn't developed by me, it was developed by Fred Nordstrom at Boehringer Ingelheim, and it's one that has been used now, and you can see in some of the publications and case studies that we've done, both at BI and Merck, those are like very, very common tests that are now being used, right? And the test is actually fairly simple. Imagine that you have a mixture, crude mixture that contains product and impurity. And let's say, for the sake of the argument, that the impurity precipitated and that you have approximately this proportion between impurity and solid. If I add a very, very small amount of solvent to that mixture, I'm going to be able to generate a saturated solution of the product and impurity, right? If I add a little bit more solvent, I'm going to still have a saturated solution if both lattices are present, but I'm going to have less solids, right? But if I look at the liquid concentration at 10 and at 20, it's going to be about the same for product and impurity. If I keep adding solvent, eventually in this case, I'm dissolving the impurity. At the moment I dissolve the impurity, I start seeing the liquid concentration drop because there's no longer the solid state independent impurity being able to replenish that model liquid concentration, right? And you can see a drop that can be modeled by simple dilution. It's a simple mass balance, right? And eventually when I dissolve the product, I should get a clear solution and a drop in concentration, right? This is a way that you can use to detect precipitated compounds, right? You grab your samples, you grab your crude that you crystallized, you put it there and do that aging for about a week and then filter, measure the liquid, right? Now, the interesting thing here is that there's two scenarios considered, right? One of them is in which you have what's called a slip one scenario. You end up having the mixture of the two impurities, but the impurity dissolves first. And the other case is when the impurity dissolves second, right? That's what happens when the impurities are either in very high amounts or in very, very low solubilities, typically the second, right? But that's important when it comes to process design. I'm going to cover that in a moment, right? Now, the problem here is most of my presentation has been on solid solutions, and this is a test about, it seems to be a test about how to measure precipitated impurities, right? But in reality, you can still recover the solids and analyze them, right? And that can tell you a lot about the solid state miscibility. So this one slip test that we did in my group with a compound that we already knew was forming solid solutions at about 10% levels. That's benzoic acid incorporated inside salicylic acid, right? And we already knew that these had solid solutions around 10%. Those were also validated using Raman, right? And we basically designed an experiment that would allow us to have a slip one scenario. What we crystallized with a slight excess of the benzoic acid so it would be able to precipitate, and then we run the slip test on that. So the one thing you can see is, of course, you still have that flat line, right? You still have that saturation point. You end up having the dissolution of benzoic acid, and then you continue having salicylic acid and it dissolves completely. Now, when you look at the solids analysis, if you were to sample the solids, centrifuge them, measure the loss on drying, I don't recommend washing in this case because you can ripen the impurities as we've measured before, right? So you basically do the centrifugation, you measure the loss on drying to know how much solvent is being evaporated, and then based on the liquid concentration, you correct for the amount of impurities that are coming from the solution into that. And you can still have a significant residual amount of impurities in the solid phase. The interesting thing here is, of course, when you have a saturated solution, you have like variable purities. In this case, the liquid phase is constant because it depends on the two solubilities. And the impurities are basically going down. They're being dissolved preferentially over the product, right? Eventually, you end up having this region in which you only have the product crystals precipitated. But look at the impurity content in the solids. They're like still like 14%, right? This is one where you can basically validate, hey, I know that there is no impurities precipitated in my sample, but I do know that the solids are still 14% impurity, even when I correct for contamination from the mother liquor, right? And at that point, it's already telling you that there's some form of entrapment within that, right? It's a form of lattice entrapment. And this all has a thermodynamic basis. If you look at the ternary phase diagram, I can define different regions. I can define one that, well, as you go more to pure solvent, you're going to have a clear solution, right? If you go all the way down, you're going to have crude solids, right? So no solvent at all. You're going to have some miscibility limit for the impurity inside the product, right? If there's no solid solution, this line goes all the way to the corner. You have some miscibility limit for the product inside the impurity. And we're going to call this solid solution of the product with impurity alpha, solid solution of the impurity with the product beta. And then the mixture of the two solid solutions is going to be the physical mixture of alpha plus beta, right? And you eventually have, if you're reading through this line without getting inside the diagram, you're basically going to get the solubility of the pure product. That's solvent and product without impurity. And then you're also going to have the solubility of the pure impurity as well as an invariant point. There's really 5 points that you need to model a system like this, right? Now, the idea is those are the different regions that we've measured. And when you run the slip test, all you're doing is you're grabbing a sample that's like solids without solvent, and you're bringing that to infinite dilution, right? Or you're bringing that to a very high dilution until you eventually reach the liquid phase, and then you stop, right? But the idea in here is you can use the same principles to model the ternary phase diagram. So you have the same slip test that I showed you before. You have the alpha plus beta plus liquid region that's right here. That's this purple region in here, right? And eventually you cross the alpha plus liquid region in which the impurity has dissolved completely. And eventually you go into the liquid region in which you have full dissolution of both. If I look at the solid state, this is the invariant point composition, right? The liquid composition is still going to be the same. It's defined by the two solubilities, right? There's a fraction of the two of them that are relatively constant. And then you have the drop in impurity levels in here. But whenever you reach the alpha plus liquid region, the one thing you can notice is that as you move up in the slip test, the impurity gets a little bit more rejected, right? So it turns out the impurity incorporation is also dependent on how much solvent you have. So that's part of like what you can see in here, right? This sort of like slow gradual drop of impurity levels in the solids that can also be validated with the ternary phase diagram, right? Now, the idea is that now you can go and you can build some simple models. If you have those 5 points, solubilities of the pure materials are something that can be obtained. Solid state miscibilities can be approximated by preparing a mixture that's alpha plus beta plus liquid, right? And the invariant point can also be obtained with that. You can actually get fairly good predictive models for, this is a case from us, a case from BI, a case from Merck. We basically publish this as a whole workflow. We provide Excel files that allow you to do all these calculations for you. If you just provide those 5 data points, right? I don't even think you need 5 data points. You only need the region that you're measuring, right? So you may need 1, 2, 3. And you can actually have the full predictive capabilities on exactly how the sleep test is going to behave to try to validate the formation of a solid solution and the thermodynamic phase diagram. And once you know that, you can now have thermodynamic control on impurity rejection. And that's part of like what I'm going to move into now, right? So let's define those 3 different scenarios. Okay, I'm going to have to wrap fairly quickly. But let's define those 3 different scenarios. Sleep 1, impurities are precipitating, but low amounts. Sleep 2, they are precipitating, and they're going to survive the product on dissolution and solid solution formation, right? First thing that you want to do is, of course, if the synthesis can be improved for selectivity, do that, right? The best impurity, the one that's never going to incorporate, is the one that's not there, right? And then, of course, the approach that you're going to take from there is going to be, are you bound to a very specific solvent system? Are you bound to a specific crystal form? And how much yield versus purity are you willing to have? Because there's usually a trade-off in there, right? Very first thing you want to do is some methods are always going to be applicable, right? So solvent screening is always very important. I know it's part of what we do now in impurity rejection. That's fine, right? But that's still going to be a very important aspect, right? That's a very general approach that tends to work. Preventing the impurity from being able to interact with the crystallization in the first place, that's a general approach, something that's very difficult to implement in practice, right? You can try to lower initial amounts, but that comes at the expense of yield. Sequestration methods like complexation, things like that, but that's also very, very system specific, right? If you don't use those general approaches, then you need to go into a very, very specific mechanism and how to address that, right? Sleep one scenario may be the easiest one to address because that's one that's thermodynamic control, right? Can you keep the impurity undersaturated? Keep in mind that the sleep test is giving you an indication of the process volume that you need specifically to make sure that that impurity is undersaturated. Add a safety factor in that, operate a slightly higher process volume than that, and you can almost guarantee that the impurity is not going to be able to crash out, right? Even with small fluctuations in crude contents, right? And that's something that trial and error is not going to be able to give you, right? So you have basically you do your typical desupersaturation profile on the product, but you always make sure that the impurity is undersaturated. And even if you don't have impurity isolated to measure the specific solubility, you can do it based on liquid chromatography area percents, right? The LCAPs and the overall content on the product. You can get an approximation based on the sleep test data, right? Post-crystallization treatments is the other option, right? The impurity tends to dissolve before the product. So you can always, if it crashes out, right, you can always add some washing, remove the impurity in there. And you want to choose a wash solvent that minimizes dissolution of the product, right? But at least you know based on the sleep test that you're having the impurity crashing out. Right? Sleep 2 scenario, that's a bit more complicated, right? You're having keeping the impurity undersaturated is impossible because you're going to be dissolving all the product, right? So one of the things you would have to do is like general methods, like can you change the synthesis, the solvent? Can you use complexation strategies to sequestrate that impurity? Because if it's precipitating, it's going to be surviving the product of any washing step, right? If everything else fails, you can always dissolve the product, do a polish filtration, remove the impurity, and then recrystallize the product. But that comes at significant yield penalties, right? We've been investigating other methods like kinetic impurity rejection, but that's not very practical at scale, right? Trying to play with seed amounts to make sure that the product is crystallizing faster than the impurity and filtering it before the impurity reaches equilibrium. But it's like it's not very practical on the industrial scale. It's more like an academically interesting. How good could it be if we could make it, right? And yeah, we're working with other ideas. You should see a bit more from us in the next couple of years on like how to deal with selective scenarios, right? But right now, this is mostly what's available, what's being done, right? Solid solutions, simplest and most effective approach. If you can change the crystal form, even if you're doing a workup step, right? You don't have to be isolating the final product while you're purifying it. If you can crystallize a different form, do that, right? This is something that nature has already found for us. What you can essentially do is like imagine that you have a form one that's able to incorporate some impurities in the lattice, right? You change the crystal form and the miscibility of those impurities is going to change. You can even expel some of those impurities from the lattice. That's something that nature has found out. That's what happens when you eat chocolate, right? Chocolate is also crystalline material, contains a lot of fats and sugars, right? And they are incorporated in the lattice of a form that's form 5. Form 5 is the second most stable form, known form of chocolate. But it's the one that we tend to go for because it tends to integrate those impurities fairly well. It has a good shine, it has a good snap. But what happens when you eat chocolate for a long time, especially at elevated temperatures, it switches to form 6 and it expels those impurities from the lattice, right? That's the blooming effect in chocolate. You can do the same thing in manufacturing, right? If you have ability to change the form of one material, even if it means utilizing an impurity to stabilize it from form, right? That's another thing we're investigating. You should be able to remove a lot of those impurities, and we actually have some case studies in the papers from before with BI and Merck that I think it's that publication where they did just that, right, and improve the purity significantly. And other methods, of course, include thermoannealing, ripening, right? So if you have solid solutions with composition gradients, and we're working on figuring out how to trigger those, you can actually have this preferential dissolution of impure regions. That's the case also with salicylic acid that I showed you the sleep test before. The impurities tend to incorporate at the core of the crystals if they're going from high supersaturations to low ones. And if you pass them through clean solvent, you're basically forming these sort of hollow crystals. You're dissolving the impure regions preferentially. And of course, there's other kinetic methods, right? There's other model-based kinetic control approaches, modeling the absorption and incorporation rates. That's from a group in Bordeaux, right? That are investigating some of these. But overall, these are mostly the methods, right? I would really recommend if you can switch the form, do that. Otherwise, thermoannealing is an option. Otherwise, playing a bit with the solvent, process volumes. You've seen a bit on the sleep test, how you can tune purity a little bit, but it's not going to make a big difference, right? Those are the things that I would be investigating, at least until now, right? All right. So to end, right, one slide and acknowledgements. If you're ever on that 6% that form solid solutions and a sleep scenario at the same time, we do have a workflow. We do have a decision diagram. That's basically a lot of what I explained today. That walks you through the different methods that you can utilize to try to deal with those scenarios, right? And that has been demonstrated with different industrial compounds. There's a part one that describes the workflow and a part two that's basically case studies from actual compounds at BI and Merck where they actually utilize these and improve the purity significantly, right? All right, so with that, I would like to thank the people working in the impurity retention and solid solutions work. We have several group members, PhD students, postdocs starting now too, and a lot of undergrads who've been working on the experiments that you've seen and some of the experiments that are coming soon, right? And collaborators, I need to acknowledge the people at BI and Merck that have been main collaborators early on, right? And more recent collaborators who are starting to work on this topic from Ella Lilly and Redwire. Funding from different sources. We still have a lot to go. There's some grants that last until 2029. There's many different aspects that we'll investigate at the same time. So you should hear more from us fairly soon, right? And with that, like, thanks everyone. If anyone needs an introduction to the industrial crystallization tool, we just published this ebook. It's fairly, I'm not getting paid for it. I'm not getting paid for like the purchase, but it's like a $40, but about 50% of the ebook is actually free as a preview. So you can actually, it's a mini course on crystallization that teaches you all the basics. So yeah, thanks everyone. I'll be happy to take any questions.
Thanks, Gerard. So, I'm looking at the chat box and we got some question coming through. So I'll just read the first one. Do you see any scope for the use of mechanochemistry in sleep test methodology?
Mm-hmm.
The APIs at different level of impurities are mixed in bone meal to keep mixtures which are suspended in solvent and analyzed to assess the impact of different level of impurity on incorporations.
Absolutely. That's what we end up doing. So we've realized very quickly that if you're just mixing the solids, like one of the things that I like to move away from is I don't want the sleep test to be perceived as a diagnostic, what happened with crystallization test, right? Because you're aging for a very long time. It's more a proactive test, right? It's one that you try to figure out the solid state equilibrium to see if that impurity has any chance of precipitating, right? By the time you collect your samples, it's no longer similar to what you had on crystallization. You've had enough time to age them. And at the moment you take that approach, it makes a lot of sense, right? To have what we tend to do is like we pre-grind the product and impurity and then mix them together, right? Sometimes we also have the shakers and we put some like stainless steel balls to make sure that we that we promote a very high surface area for contact between the solid and the liquid, right? So yes, I think mechanochemistry is going to be the way to make the solid solutions in the shortest time frame. At least it's the most promising that I've seen so far. So yeah.
Okay, so there's no more question in the chat box. Is there any question that the audience want to ask directly? Not sure if yeah, they can be unmute actually. Okay. Why don't I ask you a question, Jazz? So when you were going through your solid solutions work, the physical behaviors are very nice. The question I have is a lot of the impurity that we are very concerned about actually are at quite low concentrations.
Yes.
And how would your finding be applied to those cases? In your case, it's a case study, obviously, you pick a
Yes.
system where the balance between the two species is quite high. You know, you've got a large proportion is the so-called impurity. But if you're dealing with much smaller amount of impurity, how would the balance shift and how would it affect the behavior?
How low are we talking about?
So, well, let's not go extremely low because some of these things would go down to ppb, ppm level. But let's say if you got an impurity at about 1%.
Oh, 1% is definitely doable. So a lot of the tests that we did, even for like solid state analysis and the sleep test, had impurities that were at that level, right? Of course, the data tends to be cleaner when the impurities are at higher levels. But 1% is way above what we ended up doing. So these are the percentages that we ended up working with those industrial curves, right? So
Okay.
most of the time it was like at the 0.5, you know. The problem is when it goes into like ppm levels. So for example, in the case of acetaminophen with sulforaphane B and acetfuchsin, right? Those are very small levels. Those are ppm. And even though we can do the sleep test, we cannot measure the solid state purity that easily because the solution contains so much impurity compared to the solids to have that level of incorporation that any amount of model liquid remaining, even if you try to quantify the impurity levels, the errors are so high, right? And it's very difficult to discern when most of the impurity in your powder comes from the model liquid wetting it, right? But no, 1%, 0.1%, those are very doable on the sleep test. All of those were run with the sleep test. So.
Thanks, Raz. There are two more questions. So the first one from I'm trying to read here. Jamie Flores Vasquez is most important mechanism that promotes the formation of solid solutions. I guess that's a question. Which is the most important mechanism to promote the formation of the solid solutions?
So I think they are always gonna form. So we're trying to work like we're still trying to figure out the thermodynamics and take this with a with a grain of salt because we're still hypothesizing a lot about this, right? But the formation of a multi-component crystal is something that's thermodynamically favorable, right? Even though there's enthalpy penalties to the incorporation of impurity within the crystal, entropy likes having the impurity inside the crystal lattice, right? It's not going to allow to have something that's completely pure. It's about the same mechanism that you would see for, what is the mechanism that makes curcumin dissolve partially in water, right? It's so insoluble, but it still gets there into a certain amount. And I think it's all about reaching an equilibrium. It's the fact that there's always going to be an equilibrium between the solid and the liquid, right? And that equilibrium is not going to be 0 and 100. It may be 0.001, right, and 99.999, but there's always going to be at least one molecule sneaking inside the crystal, right? And I think it's typically impurities in solution, right, will be near or attached to the crystalline surface by basically weak interactions, right, with any exposed functional groups or basically any exposed functional groups. And as the crystal grows, they will end up inside, right? That's the line of thought.
Okay, so last questions. In general, do you think that multiple impurities incorporate independently from one another, or do they interact and affect the mutual incorporations?
There is a very recent paper from my collaborator, like Fred Nordstrom, that's I think it was published a couple of months ago. No, there could be competition in impurities. So he showed it for one of the BI compounds that there's an impurity and there's actually a solvent, and they're both competing. So you incorporate more solvent, you get less impurity, and you incorporate more impurity, you get less solvent in the lattice. So no, there's definitely going to be some competition in sites, and that's going to be one of the challenges. That's why if you run the slip test, you want to run it with the crude, right? Not just the isolated components of the product and impurity. You want to have like, the material that you're going to be crystallizing. And in that scenario, you should get a bit much more relevant information about how the impurities are interacting, right?
Okay, thank you very much. I think that's all the questions we have in the chat box. So if there's no more, then thanks again, Gerard, for an excellent lectures, and thanks to everyone else for attending.